BioRails for DMPK, a single workflow from request through data analysis to reporting results, all in one system. This is a demonstration of a brain binding assay using BioRails. I first log in as Rodney McKay. He is the chemist responsible for requesting assays to be carried out on samples. I'm making a new request which will be for sample IDs. Valid items can be requested in the assay. I'm copying and pasting a list of sample IDs and then validating them against the database. Requests can be made against multiple queues, but in this example I'll be requesting against the brain binding queue. Since this assay can be carried out using brain tissue from different species, you can select a different species for each sample that you will be testing. For this example, I am selecting mouse brain tissue. Once the samples have been selected, I can create the request. Now that the request is completed, I can sign out as Rodney McKay and sign back in as Samantha Carter. Samantha Carter is the DMPK manager responsible for managing the assays and assigning work. Each item is placed on the queue, and items from different requests can be packaged up together. We'll be putting all of the samples that Rodney requested into a single work package. First, we set the state of these items to active. This shows Rodney that they are being worked on. We can then create a new task and assign it to Janet Fraser. Janet Fraser is the DMPK bench scientist who will be responsible for carrying out the assay. The result task will contain all of the details required to carry out the task. In this example, the DMPK team leader sets the initial subject concentration for the bench scientist to follow. I can now sign out as Samantha Carter and sign in as Janet Fraser, the DMPK team bench scientist who will be carrying out this assay. Tasks assigned to Janet can be found on the dashboard. The task contains the details of the assay, and the analysis binder can be launched directly from the web page. I first import the raw data from an instrument file. The data is organized and processed by Morphet. I can check the conditions and then inspect the plate calibrations. I can now overlook the full analysis of the plate data and make decisions about how it's been analyzed. The endpoint results here are what will be uploaded to BioRails, including the charts. The table in BioRails has been set up to capture all of the endpoint data of the assay. All of the intermediate data is stored as a binder file on the server. Refreshing the page shows that all of the data has been uploaded to BioRails, including the charts. I can now log out as Janet Fraser and log back in as Samantha Carter, who will be checking up on the progress in the assay. Checking on the queue gives the status of every item that has been requested against that queue. Each active item has a link to the result task it's been tested in. All of the data presented here can be exported in a variety of formats, as required. You can also use a query to search for all the historical data for the assay. The data here can be searched and filtered as required. This has been BioRails for DMPK, Brain Binding Assay.